Right now, if you ate something sweet in the last hour, a soda, a piece of cake, a healthy fruit juice, something is happening inside your body that most people have no idea about. Not in your stomach, not in your blood, inside your liver. And if you knew exactly what was happening, the chemical chain reactions firing off right now, you might never look at a can of soda the same way again. In the next 10 minutes, we are going to walk through every single biological stage, minute by minute, of what your liver does after sugar enters your body. We are talking real science, peer-reviewed research, published as recently as 2026. No fluff, no fear-mongering, just the truth. And the truth is genuinely fascinating. Let us start the clock. Before we get to the liver, let us make sure we are talking about the same thing. When most people say sugar, they mean table sugar, the white stuff. Scientifically, that is sucrose. Sucrose is actually two sugars bonded together, glucose and fructose. The moment sucrose hits your stomach, acid splits it apart. By the time it reaches your small intestine, an enzyme called sucrase isomaltase finishes the job, and you have two separate molecules racing into your bloodstream. Now, here is where things get interesting. Glucose and fructose are not treated the same way by your body, not even close. Glucose goes everywhere, muscles, brain, heart, every cell that needs energy. But fructose goes almost exclusively to one place. Research published in BioExiv in 2025 confirmed what scientists have suspected for years. Approximately 70% of all ingested fructose is transported directly from your small intestine to your liver via the portal vein. Your liver is fructose's final destination. And the liver does not have a polite, measured response to it. Let us walk through what happens, stage by stage, starting the moment sugar arrives. The liver receives glucose via a transporter called GLUT2. This is a passive system. No energy required, no insulin needed. Glucose just flows in. The liver looks at how much glycogen, its stored form of glucose, is already on the shelves. If there is room, it starts packing glucose away into glycogen. Research from Portland Press shows that in a healthy person, roughly 50% of the glucose from a meal gets stored as glycogen. The liver is handling this efficiently, calmly, like a well-run warehouse. Now fructose arrives, and everything changes. An enzyme called fructokinase, also known as ketohexokinase, immediately grabs fructose and phosphorylates it, turning it into fructose 1-phosphate. This sounds routine. It is not. Here is the critical difference between glucose and fructose metabolism that almost no one talks about. Glucose has a built-in brake system. When your cells have enough energy, glucose metabolism slows down. Fructose has no such brake. According to a 2024 study published in Science Direct, fructose conversion to metabolic intermediates occurs completely independently of your body's energy status. There is no off switch. Fructose is processed at full speed regardless of whether you need the fuel. Think of it this way. Glucose is a car with a speed governor. Fructose is the same car, but someone removed the governor entirely. So what does the liver do with all these fructose intermediates? When glycogen storage is adequate, the liver starts converting the excess into fat. This process is called de novo lipogenesis, literally, the creation of new fat from scratch. A 2023 paper in the Journal of Endocrinology confirmed what multiple human and animal studies have shown. Fructose is a dramatically more potent trigger of hepatic lipogenesis than glucose. Why? Because fructose bypasses the most tightly regulated steps of glycolysis. By the time fructose metabolites enter the fat synthesis pipeline, your liver's normal checkpoint system has already been circumvented. The liver produces triglycerides, fat at an accelerated rate. And when glycogen stores overflow, even excess glucose joins this process. Here is the part that surprises most people. The rapid, unregulated phosphorylation of fructose by fructokinase consumes enormous amounts of AP. The liver's energy currency, according to clinical research from Pfizer's KICK inhibitor trials on NAFLD, a single intravenous dose of fructose can measurably deplete hepatic AP levels. When AP drops, a molecule called AMP builds up, and when AMP accumulates, an enzyme called AMP deaminase kicks in and converts it to uric acid, the same compound responsible for gout. You probably associate uric acid with red meat or alcohol, but sugar, specifically fructose, triggers a significant uric acid spike inside your liver. Research published in the American Diabetes Association Journal found that uric acid generates mitochondrial oxidative stress. Essentially, it damages the liver's power plants. It also inhibits a key enzyme in beta oxidation, the process your liver uses to burn fat. So not only is your liver making more fat, now it is also less capable of burning it. More fat being made, less fat being burned, both happening simultaneously, in the same organ, in the same hour. Your liver is not a storage facility, it is a processing hub. So what does it do with all this newly synthesized fat? It packages it into particles called very low-density lipoproteins and ships them into your bloodstream. Think of VLDL as the liver's delivery trucks for fat. According to a study on VLDL kinetics, high-sugar diets specifically drive the liver to produce more of the larger, triglyceride-rich VLDL-1 particles in healthy people. These are the particles most associated with cardiovascular risk. When those trucks unload at your fat cells and artery walls, that is where long-term damage begins. Here is a plot twist that will make you rethink everything. What if you avoid fructose entirely and just eat a high-glucose diet? Maybe a bowl of white rice, a large potato, lots of bread, no added sugar, no corn syrup. Are you safe? Not entirely. A landmark study published in Nature Communications and updated with new data in March 2025 discovered something remarkable. Your liver can convert glucose into fructose internally through a pathway called the polyol pathway. When blood glucose is high, an enzyme called aldose reductase converts glucose into fructose right inside liver cells. This endogenously produced fructose then kicks off the same cascade: the fat synthesis, the ATP depletion, the uric acid spike, all of it. In studies, mice that were genetically unable to metabolize fructose were completely protected from fatty liver, insulin resistance, and weight gain, even when fed large amounts of glucose. Remove the fructose metabolism pathway, and glucose alone cannot trigger the full metabolic disaster. This means the real culprit is not just the fructose you eat, it is the fructose your liver quietly manufactures from too much glucose. The liver is not a passive victim of your diet, it is an active participant in its own metabolic fate. One sugary meal does not ruin a liver. The body is resilient, it recovers. But repeat this pattern every day, multiple times a day, and the liver begins to lose the fight. Chronic high sugar intake leads to a condition called metabolic-associated steatotic liver disease, the new name for what was previously called non-alcoholic fatty liver disease. According to a 2024 systematic review in Frontiers, this means more than 5% of hepatocytes, liver cells, are packed with fat droplets, with no alcohol involved at all. The progression follows a predictable arc. First, steatosis, fat accumulation, mostly silent, no symptoms. Many people have it without knowing. Second, steatohepatitis, inflammation added to the fat. The liver becomes injured, enzymes leak into the blood, doctors find it on routine blood tests. Third, fibrosis, scar tissue begins replacing healthy liver tissue. Fourth, cirrhosis, severe, permanent scarring, liver function declines. And research published in BioAudiv in 2025 projects that by 2050, nearly 1 in 4 liver transplant patients will be there because of NASH, sugar-driven liver disease. Uric acid plays a significant role across all of these stages. A 2024 paper from NE confirmed that elevated serum uric acid is an independent risk factor for NA-FELD, activating the NL-ARP3 inflammasome, a key trigger of liver inflammation. And the gut is involved too. Research published in the Journal of Clinical Investigation in April 2025 showed that high-sugar diets alter gut bacteria composition, increasing bacteria that produce a compound called DCA, which blocks the liver's FXR receptor, the master controller of bile acid, fat, and glucose metabolism. Your gut, your liver, your microbiome, all connected, all disrupted by the same thing. Now, before you spiral into existential dread, here is the part that actually matters. The liver is one of the most regenerative organs in the human body. Research consistently shows that reducing dietary sugar, even for as little as 7 days, measurably decreases liver fat content. A clinical trial currently underway is testing exactly this, replacing fructose with starch for just 1 week to measure liver fat changes in patients with early-stage fatty liver. The science tells us a few clear things. 1. Liquid sugar is the most dangerous form. Sodas, juices, energy drinks, fructose dissolved in liquid absorbs faster, hits harder, and bypasses satiety signals almost entirely. 2. Fructose from whole fruit comes packaged with fiber, water, and micronutrients that slow its absorption dramatically. An apple and a glass of apple juice are metabolically very different things. 3. The liver recovers remarkably fast when you give it the chance. Low-carbohydrate diets have been consistently shown to reduce hepatic de novo lipogenesis within days. You do not need to eliminate sugar from your life. You need to understand it well enough to make conscious choices. Because now, you know what happens in that first hour. You know about the fructokinase cascade with no off switch, the ATP drain and the uric acid spike, the fat factory switching on, the VLDL trucks heading out into your bloodstream. The liver quietly converting glucose to fructose when no one is watching. That knowledge is more powerful than any diet trend or supplement. If this video changed how you think about sugar, even slightly, share it with someone who needs to see it. Drop a comment below. What did you eat in the last hour? I promise I will not judge, but your liver might. Subscribe if you want more videos that go this deep into the biology of everyday choices. We do not do surface level here. See you in the next one.